डर लग रहा है क्या? डरने की ज़रूरत नहीं है। ऑन 27 मार्च अपना बायोलॉजी का पेपर है ना, तो बायोलॉजी के पेपर के लिए हमको इस समय मोस्ट ऑफ द इम्पोर्टेंट क्वेश्चन चाहिए। सो योर टीचर फ्रॉम पीसीबी बाय अंकित सर, मैं आपको वो सारे टॉपिक्स देना चाहता हूँ जो टॉपिक्स मोस्ट ऑफ द इम्पोर्टेंट प्रायोराइज्ड, इवन प्रायोरिटी मैं उसी को ही दूंगा बिकॉज ऑल द क्वेश्चंस आर कमिंग फ्रॉम देयर। सो डोंट वरी, get through it and try to focus on small to small type of the questions, try to make a thinking based questions, और इसी के साथ आप इन सभी टॉपिक्स को धीरे धीरे रिवाइज कर जाना। 100 एंड श्योर 80 to 100 के बीच में मार्क्स आपके रहेंगे। इवन इफ यू डू मोर देन यू विल गेट 90 प्लस। सो डोंट वेस्ट योर टाइम, लेट्स गो टू द वीडियो। चलिए चलते हैं।
So this one is the first chapter in which transfer section of anther and its layer name and function is present. Even you see microsporogenesis and megasporogenesis, artificial hybridization, embryo sac development. You will see the types of the cross pollination, means pollination you have to read, and digot and monocot embryo, the embryo development. You also have to see the development and also diagrams. Try to create a diagram, try to practice the diagram in your notebook so that you will easily understood this chapter, and each and everything is in the description so you can download. In this second chapter you will see the testis and its working with seminiferous tubule, accessory duct flowchart, spermatogenesis and hormone control like in LH and FSH working, human sperm diagram, oogenesis and hormonal control, menstrual cycle, placental development and embryonic development. In this chapter also you practice the diagram. It is very important for you. In this chapter, contraceptive methods like IUDs, pills and surgical methods are used, amniocentesis, read ART and all its processes. It also includes ZIFT, ZIFT, artificial insemination, IUI, ICSI. Practice all of them. This fourth chapter, principle of inheritance, consists of incomplete dominance and codominance, in which you also see multiple allelism like ABO blood grouping. You also practice the test cross with Punnett square, and even you understood about T.H. Morgan experiment on Drosophila like linkage and crossing over. Pedigree analysis, sex determination in birds, human, honeybee, and even chromosomal and Mendelian disorder. You must have to understood all of these. Molecular basis of inheritance consists of DNA structure and double helical model, mostly coming, even Chargaff rule, Hershey and Chain experiment, DNA replication, transcription, translation, silent features of genetic code. Even you practice the lac operon and the question coming from the lac operon, human genome project and the goals of the human genome project. Even you also see the DNA fingerprinting. So each and everything is present in the description. You also have to be downloaded from there and understood. If we see about the evolution, homologous and analogous organ questions and with example will come. Question first arises. Miller's experiment diagram, industrial manualism also have to be understood. Hardy-Wingber principle and numerical, natural selection graphs, adaptive radiation in which Australian marsupials and placental mammals must be there, and the last one is human evolution must understood. Now if we see for the human health and disease, all diseases and its causative agent must be learned. Plasmodium and life cycle diagram banakar, after that you have to practice it. Immunity in which B and T cells, humoral and cell mediated immunity, barrier and innate immunity, active and passive immunity, even in antibody diagram and type like GAMDE, AIDS and cancer, and even drugs. For the microbes and human welfare, microbes uses in different type of the reason where the microbes are used. We go for the sewage treatment, so sewage treatment plants must be practiced. For the biogas plant, it is very important for the biogas plant which gas is produced and which microbe is used. And the last one is the biocontrol agent, that must be also important. This biotechnology unit consists of restriction enzyme and its naming and working, must practice it. Gel electrophoresis, very important. PCR, polymerase chain reaction, must be read. PBR 322 diagram with all labeling, याद करोगे याद हो जाएगा. Insertional inactivation, micro injection and ballistic gene gun, bioreactor and downstream processing, very important. All things are present here. In this chapter, Bt crop, Bt cotton, crygene and protein must be important. RNA interference is important. Human insulin and diagram, that is also important. Gene therapy, transgenic animal and biopiracy, this is important. You have to practice the diagram day by day, and also have to practice the names and learn the names very seriously. For the next chapter, organism and population, you must practice age pyramid, growth models, population interaction and examples, brood parasitism, and even population density, in which nt plus 1 and nt plus b plus I and minus d plus e. This must be very important. Each and everything is in the description. For the next one, we see for the ecosystem, primary and secondary productivity is very important. Decomposition is important. Ecological pyramid is important. Food chain is important, in which different type of the food chains and different ecological pyramid must be practiced. You also must practice which one is inverted, which one is upright, and which consists of different type of pyramid. The next one is biodiversity and its conservation, in which species area relationship and its diagram and graph. Loss of biodiversity for the reason which is evil quarter. Biodiversity and its conservation, broad and narrow utility and ethical reason, and even ex situ and in situ conservation with diagram. So here overall I'll tell you the topics. Thank you for sorting this video. Like, share and subscribe, and also have to understand and practice well. Thank you.